I was asked to talk about the way that we can look at individual patients' genes and see if we can use that to improve the way we make a diagnosis or the way that we treat people. And my own medical specialty is heart disease, so I'm going to talk particularly today about the way we use genes and genetics in heart disease, but I hope that what I illustrate would hold just as well in other areas of medicine. And given it a title, I wondered whether to talk in general about common genes that we all have in heart disease, but it turns out that's still medicine that's for the future. And what I'm going to talk about today is a particular area of research that's been done over many years in Oxford. A lot of it supported by the BRC that I think now does impact on the way that we treat and look after our patients. And I hope it will illustrate how we do this sort of research and where it takes us to. So I think the right place to start is to consider the question, how do we consider how much of a disease is caused by the genes that you have and how much is caused by the environment that you live in? And you'll know about the nature-nurture debate, it's much written about. But the answer is it's different from one disease to another. And I think the easy way to consider it is on a spectrum where I've got some conditions here. And this is the end of the spectrum where everything is very much due to genes. And this is the end where everything is very much due to your environment, what goes on about you. So you will be familiar with the fact that there are conditions that are inherited in families that are caused by a spelling change in one gene. And so common conditions that we know about are things like Huntington's disease or cystic fibrosis. And here, as you likely know, there is a gene for these conditions, and if you inherit it, you'll get that condition. So this is the very strong genetic end of the disease spectrum, and these conditions tend to be quite rare. At the other end, there are conditions that even I, as a geneticist, would consider have no gene contribution. Not very many. I put traffic accidents. You're more likely to die of a traffic accident if you live in Istanbul than if you live in Oxford. But actually, you're also more likely if you're a risk taker. And I think risk-taking is quite a genetic trait. So I've given it a little bit of genetic influence here, but mostly it's environmental. Now, if you ask me about the diseases that are common and important, that lots of people have, are you going to get diabetes? Are you going to have a heart attack? Do you have high blood pressure? Are you going to get forms of cancer? They sit in the middle of this spectrum, where there's plenty of genetics going on, but also plenty of environmental factors. So if you're talking about heart disease, there are lots and lots of different genes that can make you more or less likely to get heart disease, and a lot of environment factors. Do you smoke? Are you overweight? Are you taking enough exercise? So there's a lot of research that goes on in the genes that make you more or less likely to have these common diseases. But at the moment, the things that we find from that work is not very easy to use at the individual level for the patient. And it's complicated because there are lots of genes that can affect your chance of having a heart attack, and there are lots of environmental factors. So if you come and see me in clinic and you've got one of these conditions, I won't be testing your genes. We might be in the future, but for now we're not. So if you want to ask, where does looking at genes or looking at the genome, which is all of your genes put together, really impact on medicine, then at the moment, it's at this end for the rare diseases. So I'm going to pick one rare heart disease that's due to a disease gene, a particular spelling mistake in a particular person, and talk you through how we use genetics to try and improve the way that patients with that condition here, but actually across the world, get looked after. So the condition is one with a rather uncomfortably long name, but it's a pretty simple name, and it's one I've worked on all through my career. Hypertrophic cardiomyopathy. I probably will call it HCM for laziness during the talk. Hypertrophic just means thickening. Cardio is heart, and myopathy is muscle. And it's a condition that's characterized by thick heart muscle. So I've got a couple of images here to show you hearts and how we look at them. And the one on the left is a nice, normal, healthy heart. These are images that come from a magnetic resonance image scan. And it's a fantastic scan that we have now that gives us way more lovely images of the heart than we've ever had before. It's completely safe, so it's a lovely tool for research. You can do it as many times as you like. There's no radiation, and we get fantastic pictures. So this just shows you a normal person's heart beating away. It beats once every second, sitting in the chest while everything else is rather still and doing an awful lot of work. And you'll find out later that a lot of my talk is about the work that the heart does. So the blood flows in here. This is actually coming from the lungs. It goes into this reservoir chamber through this little valve, and this is the pumping chamber, and then it pumps it out, and you can't actually see the artery coming out. So this is a normal heart, and you or I would have a muscle thickness there of probably under 1 centimeter, maybe about 10 millimeters. So then the scan on the other side is a patient who has this hypertrophic cardiomyopathy. So here, the muscle's too thick. It's about 2 centimeters, so it's thicker than normal. And in fact, if you look at this chamber, the pump, it's very muscle-bound. This is like it's been to the gym working out. And it actually blocks itself off. Here, if you look at the pumping, even when the heart is contracted, there's plenty of blood left. But this one is sort of closing down on itself. This isn't a terribly bad pump. And if you had it, you would probably find you could do normal activities. You might not have any symptoms. You might well not know that you had it. And that's the problem, because it does cause other trouble. So these are some of the features of the condition that you need to know to understand the talk. So it is inherited in families. This is a genetic condition, and we've known that since it was first described in the 1950s. If someone's got it, you'd usually find either their mum or their dad had it too and have passed it down. And if they've got it, usually half of their children will have it. The thickening comes on usually in sort of teenage years, sometimes later. And although I said genetic conditions are uncommon, it's not really rare. So 1 person in 500 has this condition. So if you go to a GP surgery, they'll probably have a handful of people with it, but they probably won't know who they are, because most people with it haven't been diagnosed because the heart works pretty well. And the whole reason it's medically important is this, that this is a condition, this is the commonest of the many conditions that can cause young people to die suddenly. So if you read or hear in the press of a young kid who's died suddenly playing football, this is usually what they've died of. So it's a horrible condition for the families involved, and one that you would really want to become better at treating. And I can illustrate this because it happens in the public domain, and one of the recent ones you will remember, which actually had a good outcome, was Fabrice Muamba. So he, a footballer, highly talented, highly athletic. He had this condition, but his heart could perform perfectly well to be a footballer, until out of the blue, you get a catastrophic electrical event and the heart goes haywire and stops pumping, and you die. And he was only saved because all of the doctors and the team were there, and they got his heart going again. But usually, if this happens in other circumstances, you die suddenly. And we think somewhere between 500 and 1,000, mostly young people, will die each year of this condition in the UK. So it's not rare. It's not a tiny number. If it happens in families, as you can understand, it's particularly horrible. This is a pedigree, and the way we draw these is we use squares for men and circles for women. We use this dark color, meaning it's got the condition. And as you can see, it comes vertically down the generations, and roughly half of the kids in each family get it. And we use these lines for someone who's died of it. So at its worst, it's a shocking condition. This is a woman I know well who lost her dad, her husband, her daughter, and her son, all dying suddenly, years apart, because of this condition. Other bits of the family, people don't die so much. There's a pattern here we don't fully understand. So not very rare, but not very common, and very serious, and difficult to diagnose. This is a very big family tree. Pretty uncommon to sit in clinic and draw a family tree like this. And the reason it's big is that you can, with a big family, go out and find the gene. So when I started this work, we didn't know what caused it, but this family, and it had branches on different continents, helped us find one of the genes for the first time. So I like to show this slide also. This is another family. The notation is the same, males and females. I like to use it to tease my junior colleagues. This is an ultrasound of the heart, which is a lot less easy to interpret than that beautiful MR image. And if I put a junior doctor on the spot, how much thickening is this? Is it enough to decide that it's HCM? It's a difficult scan to interpret. And so while they struggle with that, I then wanted to see if the rest of the audience is struggling with this, because you probably should be. This is the patriarch. He's got an awful lot of kids. Got a family here, about 12 kids, another family here. Oops, now has a family with granddaughter. So before that makes you really uncomfortable, you read the small print. The patient, Alice, is 2 and is feline. So as it turns out, this is a very common disorder in cats. And cat breeders do this sort of thing, and it's not considered improper, so we can relax about it. So this is actually commoner in cats than in people. It's the commonest heart diagnosis a vet ever makes, and it causes cats actually to have a different catastrophic event. They have a blood clot in the heart that blocks the artery to the legs and die suddenly. And one of my cats actually, after I made this slide, died of this condition a couple of years ago. So it's common in cats. But again, we spent a lot of years of my life mapping cat pedigrees to see if it was easier to find the cat gene than the human gene. And we needed to do the mapping because at the beginning of this work, we had no idea what would be going wrong in the heart. We knew that within each family, there would be just one gene change messing up one gene and therefore one protein. But the heart's quite complicated. If you strip it down, it's a muscular pump. The muscle is a network of cells that all join together and contract together, and each cell is long and thin. But the molecular machinery within the cells is very complicated. The heart has to take signals from the outside, that's out here, an electrical signal to tell the heart cell to release ions, calcium ions, into the inside. It has to have a molecular motor. The motor in the heart is a sliding filament that slides like so. It has to have energy. It has to have these little things called mitochondria that make the ATP, which is the energy. It has to talk to the nucleus. There are thousands of proteins in the heart, and we did not know which one would be abnormal. But you can, with a big family, narrow down the gene and find out where the trouble is. And I can summarize about 10 years of my research life by saying the trouble turned out to be here in the molecular motor. And I can show you that in a slightly expanded form on my next slide. So this is a complicated motor. There's one strand called the thick filament, one strand called the thin filament, lots of different proteins, and their job is to burn ATP, the energy molecule, and contract by sliding past one another. And it was completely unexpected, but it turned out that different families had different spelling mistakes in different genes, but they were all here in this motor. So in terms of the scientific discovery, I reckon that equates to about one line in a medical student textbook. But I'm very proud of that line, and it was what got me my chair in Oxford, so I'm not going to knock it too much. So instead of being a condition of unknown origin, it's now a condition that's caused a disease of the sarcomere, this contractile machine here. But what you need to know, because you're here to think about patient-based research in Oxford, the biomedical research centres, how do we use this to diagnose our patients better, and how do we use it to come up with a new treatment? And I'm going to briefly touch on both of those. The problem we have is that patients crop up with this disease, let's say here, maybe they have a collapse, but they get away with it, they don't die, or they have symptoms, or it's found by chance. And we suddenly know that there's a lot of relatives who are at this 1 in 2 chance of having the condition. So a lot of concern, and a lot of very expensive clinical tests. The scans we showed you are expensive, and they're quite hard to interpret. I can't always tell, particularly for youngsters, if they've got it or not, and I can't comment on the ancestors who've already died. So the genetic testing might have advantages. And it's something that we did with the BRC money. We took the genetic testing out of my research lab into the NHS lab in the Oxford University hospitals and adapted the technology and made it available and have now provided it as a service for the NHS in the UK. And it's now quite easy with one of these families to test the 10 or so different genes and the 2 or 3 or 4,000 or so different spelling mistakes that we know can exist to find out what's going on. And later we might, perhaps in discussion, talk about the fact that we can do way bigger genetic tests than that. But this sort of one can now be done for a couple of £100, and it's relatively quick. And when you do that, you get clarity. So when we can find mum's mutation, I can find out, yes, her son's got it, yes, her brother's got it, but other members haven't. And if they haven't got it, they're not going to get the condition. And because it's quite easy once you've found that gene change to test other relatives, we can then start going through the family and going, well, where else is this condition present? And if you don't have it, you and your descendants are out of the frame. So I now know it's not on this side of the family. There's a whole bunch of people that don't need to come up and have scans. And they don't need to have repeat scans every year while they grow and every 5 years through their life. So if you have got it, you now need cardiology care. And if you haven't, you can go. And if you haven't got it, your descendants haven't got it. So this turns out to be cost-effective medicine, because we did an analysis here in Oxford that did a health economics analysis and showed that actually you can save money by testing the gene rather than doing the scans. And in fact, the cost you pay to save a life, and I'll come back to that, is very affordable. And interestingly, it was that drove this into new clinical guidelines. So as you may know, doctors are quite governed now by guidelines. We practice, we get together in a big consensus, a group of people, we decide what the best evidence is, and we say, this is how you should treat your patients. And so now, just for the first time this year, there was enough evidence to say, look, you should look after these families by looking at their DNA than looking at their hearts. And that was on the basis of Oxford work, but interestingly, particularly the health economics analysis, because in the NHS, money is very important. And if you can show that it's cheaper or affordable, it will get adopted. So this is now a standard of care. And class 1 indication means you should do it. In America, it means you'll get sued if you don't do it, but you'll get reimbursed if you do. So this is now a very active thing worldwide. And it turns out to be a very active thing in the veterinary community as well. You can take your pussycat along and get his or her DNA tested as well. So if you're going to do that, you've got to be sure that it's useful. You've got to be sure that your patients and their families are going to turn around and go, we're better off knowing than not knowing. And that means you've got to be able to do something about their problem. And it turns out we can, though not yet perfect. When we find a family with lots of people with the condition, usually most of them look absolutely fine, and then there'll be the odd one that we can tell has an unstable heart that could put them at risk of that fatal collapse, the Fabry-Miranda collapse. And it is definitely a subset. What do we do when we find them? Well, the one thing we have at the moment that absolutely works is a complicated thing, and that's not a tablet treatment, it's not a biological treatment, it's a device that you may have heard about that we call an implantable defibrillator. So this is a bit like a pacemaker, but quite a lot more. So here's an X-ray of a patient. They have a box under the skin here, which is like a pacemaker, which you can feel, but it's sort of deeply buried under the skin or the muscle. One wire around to the bottom of the heart, one wire around to the top of the heart. And this machine monitors the heart rhythm day in, day out. And here's a real trace of a patient of mine who's having nice, regular heartbeats, and then suddenly goes into this life-terminating rhythm change. So this patient will for sure die if a doctor doesn't get to them within about 5 seconds, or a device. And the device waits about 5 or 6 seconds, and then boom, it gives the same shock that you'd get with paddles if you had a resus in the ER, and the heart starts up again. And my students sometimes say, that's a long time to wait. Is it charging up? Is it not very smart? And the answer is neither of those things, but you really, really want to be unconscious when this thing goes off. And 5 seconds is long enough to be unconscious, but not to perish. So I did one of these Oxford Sparks animations for school kids, and I did it around a patient of mine who's a very gruff, uncomplaining forklift truck driver. And we'd finished our regular checkup, and he's out the door. He said, oh, I did have a bit of a spell a couple of weeks ago. And I go, what happened? He goes, I don't know, not much. I don't remember it. So we take our computer and we interrogate the device, and he was in his forklift truck doing his shift, and he'd had one of these, so he'd basically died. It turns out if you slump in a forklift truck, it stops driving. He'd had his shock, he'd come round, and he carried on his shift. His colleagues didn't notice, he didn't notice, he didn't tell his wife about it. So at best, They're very good. They save lives. But as you'll appreciate, it's a very last-ditch sort of medicine. You wait to the point that someone basically perishes and bring them back. And we would like to be smarter than that. We'd like to say, we know about the genes, we know about the proteins, can we not now fix the molecular defect? And I'm going to show you just a couple of slides about that. We now have to turn away from the patient back to the laboratory. Oxford has very powerful basic science laboratories. We have very clever colleagues who can look at the structure of the molecules. These are, this is the myosin motor, this is the actin molecule it binds to. These are the actual molecules that drive the molecular motor in the heart. And in blue here are all our changes that cause this condition. And in red, changes that cause an opposite condition. We can make these proteins. We can take the gene from, let's say, a human DNA and clone it and express it in a bacteria and make the protein and study them in test tube experiments. And I'll illustrate the only one that's sort of visual. We can stick the motors down on a surface so they don't move, and then put above them the muscle filaments that they have to slide around and label them so we can see them move, and they whiz around like this. So these are actual molecules of muscle, heart muscle, that we've made in the test tube whizzing around. And we do that to say, so what's wrong with the motor? Is it too fast, too slow? What are these mutations doing that are making it not work normally? And the lesson that we got out, which was at first confusing, is lots of things go wrong. Mostly they go fast. Some of them still generate force normally, and some generate less force. And for a while, we were very hung up on this because it wasn't very consistent. And then we thought, well, the one thing that all these mutations do is that they waste the amount of energy that the muscle uses because it's burning along fast and generating either normal or below normal force. And that turned out to be very important, and the light bulb went on, and we realized that there must be other genes that did this. And this is a summary of processes involved in heart muscle cells for either using energy or sensing it or making it, and you don't need to worry about the details, but we've since shown, because it's now much easier to find these genes, that mutations in this or this or this process all cause the same cardiomyopathy. So we've been quite excited that this was all about energy in the heart. And the reason we like that is that there are good reasons to think you might make that better. Whereas if it's a mechanical problem, if you've got a car engine and there's a mechanical problem, fixing that's really hard. But if it's a fuel supply-demand problem, that's much easier. Then we go back to the patients, and we go back to our MR scanner that does the pretty images, because it can also measure the biochemistry. We can measure how much of this fuel molecule, ATP, there is in the heart. This is extremely high-tech, but we then had to become low-tech because we wanted our patients to exercise. But they can't move, and we can't use a bike, and we can't use a treadmill because it would fly across the room and wrap around the magnet. So it comes out as rubber bands and pulleys. So if you volunteer in one of my subjects, in one of my studies, rather, this is what you'll be asked to do. It works. And when we do that, it turns out that patients with this condition have really, really low energy stores in the heart, and when they exercise, they plummet down, and they don't recover in the way that you or I would. And it turns out that the other thing you need energy for in the heart, apart from contracting, is sorting out the electrics of the heart. And this is why they get into trouble. They don't have enough ATP to drive the ion changes, and the electrics goes haywire. And that's why the youngsters who die suddenly often do so when they're doing sport. So the last slide before one sum up one says, can we do anything about it? Well, we think we can. So it turns out people in drug companies making drugs to treat angina have tried to make drugs that change the energy supply in the heart for many, many years. And they made one called perhexline 20 or 30 years ago, which works quite well, but doesn't get used for angina because better things have come along since. So we did a small BRC-funded mechanistic study in Oxford and other centres where we took patients and we asked how many symptoms they have. This is a measure of symptoms. And we did a before and after. And the dummy pill, they have the same level of symptoms, which is moderately bad. And the patients get radically improved on the perhexline. And this is actually the best result of any treatment that's ever been used in this condition. And my great excitement is that I think this same treatment might actually stop the patients developing the condition in the first instance. And so we're now doing, difficult to get funded because it's off patent, but we could talk about how we do that, a large multinational, what we call endpoint trial, giving it to patients to see if they do well, stay alive, and stay out of hospital. So if I finish up now, I hope what I've shown you is that with the BRC and the research goes here, we're now pretty routinely testing for genes that cause inherited diseases. And in this particular one, we use it to ask who's at risk. If we know that at risk, we have devices, but we hope that before long we'll have new medicines. Thank you very much.